Hi there. In this final video for the second part of the course, we'll discuss modern genetics, of which the primary goal is either figuring out what genes do or figuring out what genes are responsible for a particular phenotypic trait. One type of trait that is studied a lot is disease. So much of what you'll read about genetic studies is to find a gene responsible for a certain disease. So we'll talk specifically about the method called linkage disequilibrium. This is the method that predominated to approximately the mid-90s. Since the mid-90s, with the advent of our ability to sequence larger portions of genome more quickly and more cheaply, people have been doing genetic association studies. We'll talk about what is being measured when a genetic association study is being performed, and that's a SNP, a single nucleotide polymorphism. And finally, we'll discuss how SNPs can travel together in groups, which is also to say that portions of the chromosomes are inherited together as a block, and these blocks are referred to as haplotype blocks. Diseases are frequently caused by mutations, and so it's reasonable to discuss several different classes of mutations that can occur. On the left here, we see a wild-type protein with a particular function that's indicated by these red rays emanating from one of the protein termini. Proteins can lose their function due to mutation in a variety of manners. One, there can be a single point mutation, the change of a single amino acid, that is going to abolish this activity. There could also be a truncation event where an entire portion of the protein is lost, very frequently due to a mutation which causes a premature stop codon. Thirdly, a gene can be deleted entirely, producing no protein. Less common, but still very medically important, are gain-of-function mutations. We recently discussed RAS as a protein which can have a mutation which will cause it to signal even in the absence of a growth ligand, and so mutated RAS can predispose to cancers. That's an example of a gain-of-function mutation. A third type of mutation is called a conditional mutation. In this case, it's a temperature-sensitive mutation. In this case, there's a mutation in the internal portion of the protein which causes it at normal physiological temperatures, 37 degrees Celsius, for the protein to misfold and therefore have no activity. But perhaps at lower temperatures, the protein can still fold properly and therefore have activity. Conditional mutations in proteins can be a useful experimental tool in the laboratory, but is not of any particular advantage to any particular individual. The primary way of doing genetics up until the mid-90s was a method called linkage, in the shortest version, or linkage disequilibrium. This approach relied on the analysis of large families, large pedigrees, where a disease occurred very frequently. These large pedigrees or families could sometimes include several hundred people, with many affecteds. The diagram here records relationships between individuals, indicated by lines joining them, their gender, indicated by shape, with squares being males and circles being females, and the presence or absence of a disease. The presence of the disease, in this case deafness, is indicated by a blue color. A healthy phenotype is represented by a white color. Once you have a pedigree like this established and a sample of the genetic material from each of these individuals, people would typically look at about 200 or 300 locations in the genome and would try to identify a portion of the genome that is present in all the affected samples, but not any of the unaffected samples. At those times, looking at approximately 300 different loci in 100 to 200 people was a Herculean effort. Nowadays, you could do much more in-depth study in a few weeks. But to explain the advantages of the methods used then is that, number one, for this disease, deafness, you can assume, since it occurs in a family, that all the people who are deaf here are deaf because of the same reason, so there's probably a single mutation causing it. That gives you some statistical power. If you just take deaf people at random from the population, they might be deaf for different reasons, and so the genetic signal might be expected to be much weaker than in this case. One of the disadvantages of this type of approach is that with relatively small numbers of people participating, there've only been a relatively small number of crossing over events on the chromosomes, and so that one might not be able to localize any particular mutation or gene very accurately. If you think about checking 300 different loci in a 3 billion base pair genome, each of those chunks of the chromosome is going to represent about 10 million base pairs. So you might be able to localize a disease-causing gene within 10 million base pairs. It could still be a large amount of work to get from that particular chunk of DNA to the actual disease-causing gene. As sequencing methods got cheaper, experimental designs evolved in no small part because large pedigrees with certain diseases don't exist very commonly for a large amount of diseases. So the approach nowadays is called association study. And in particular, there's an abbreviation called GWAS, which is Genome-Wide Association Study, in which an entire genome is scanned, looking for associations, correlations basically, between a particular genetic variant and a disease. So in this case, we would take a cross-sectional cohort of the wider population, divide them into diseased and healthy groups, and look for portions of chromosomes which tend to correlate with a disease status. In this example, the yellow fragment of the chromosome is intended to be disease-causing, and you'll notice that there's one over here in the healthy cohort. And this highlights a complicating factor of this type of study, and that is incomplete penetrance. You may have heard about that before. You might have the gene, but because perhaps you were not exposed to certain environmental stimuli, or the rest of your genetic background modifies the disease gene in a certain way, you might not manifest the consequences of the disease even though you have the disease-causing gene. In contrast to the linkage disequilibrium studies, where perhaps 300 people were studied, modern association studies can now involve tens of thousands of people. And that is also a Herculean effort. And it usually involves patient recruitment at many sites, so coordination among many hundreds of investigators. 10 or 15 years ago, people would have expected that those kind of numbers would have been sufficient to identify many, many more disease-causing genes than we have actually found. So in that sense, the modern genetics is a bit of a disappointment, and also a bit of a mystery. If you can't find the genetic components of these diseases with that kind of studies, where is the missing heredity? Why are you not seeing it in the genome? We can discuss this issue a bit in class. So here's a concept you need to understand thoroughly, and which has confused some students in the past. When people do the genome association studies, they don't sequence their whole genome. There's no need to. Because humans are a relatively young species from a generational perspective, not every base pair is different between you and I. Between any two human individuals, approximately 1 out of 500 base pairs might differ. But it's not 1 out of 500 randomly. There are certain locations, call them hotspots if you will, where people tend to be different. These are referred to as SNPs, or SNPs are short for single nucleotide polymorphism. So that's a single nucleotide, and polymorphism means can be two different forms, can be a G in this case or an A in this case. And although there are 4 bases, and you could conceivably have 4 variants at any particular location, again because we're a young species, we haven't had enough time yet to reach that. And in fact, most of these single nucleotide polymorphisms exist in only 2 varieties in any appreciable amount in the human population. Now, SNPs can be thought of as common mutations, right? If this is the wild type sequence and someone has a mutation in this second base, the G turning to an A, that will be called a mutation. So the distinction between a SNP or mutation is blurry, and generally people use a rule of thumb. If a base pair change, like G to A, is present in less than 1% of the population, it can be referred to as a mutation. And if it's prevalent, if it's 10% of the population or 20% of the population, is considered a common variant or a polymorphism. But because, again, humans share a genetic history, I don't need to sequence all 500 base pairs to understand what the chromosome looks like around here. I only need to sequence the single base, and that's going to tell me what the other 499 bases look like. If you have a G here, you have one set of 500 base pairs. If you have an A here, you have a different set of 500 base pairs here. And I'm speaking on average, of course. In fact, it's even more pronounced. Here is possible results, hypothetical results from an association study. We have healthy control population, we have a diseased population, we have genotyped many SNPs across a chromosome, and we see here that in this region, healthy people have only a set of 4 SNPs that occur always as a block, and the diseased individuals only have a different set of SNPs, but they're all different at all 4 locations. This is a consequence of the fact that, genetically speaking, these loci are close enough that there has never been in this population a crossing over at this point in all of human history, all people for all time. And so this segment of the chromosome is inherited as a unit. Now, we don't know which of these SNPs are causing the disease, and we also, even if there was one that was unique, that wouldn't prove that that's the disease-causing mutation, only that the mutation that's responsible for the disease is close to that polymorphism. Now, if we only differ once every 500 base pairs and these 4 differences travel as a group, then you might be able to say that this region of the genome represents about 2,000 base pairs. In fact, the blocks that we inherit as a group on average in humans is between 20 and 40,000 base pairs. And this might indicate that we have not chosen to identify every SNP within this block. But because we only have to sequence a single base pair, it's possible to check 10 million bases, 10 million different SNPs or haplotype blocks to get a fairly complete picture of what a person's genome might be. But emphasizing the fact that this is an assumption that works for common genes and common diseases. There's always that rare mutation which only exists in a very small number of people, which you have to use more in-depth methods for discovering. This slide reinforces these points, showing that for a particular region between SNP 4 and 5 and between SNP and 12 and 13, human chromosomes only come in 2 varieties, in this case pink or blue. But the flanking regions, there's no particular correlation between what you find in the flanking region. Again, this is called a haplotype block and can typically represent 20 to 40,000 base pairs. Haplotype blocks are discovered by taking a lot of individuals, looking at sequencing or identifying a number of SNPs at various locations, and then calculating correlation coefficients between any 2 SNPs. And so right here you can see here is one haplotype block which corresponds to this region of the DNA right here, and that there's also high correlation among SNPs here, and so that there is a second haplotype block located between these 2 markers. So to a first approximation, if I learn what's at any one of these SNPs, I will know pretty much what's going on at all this location. And in the last slide, we talk about nature versus nurture, or genetic versus environmental influences. And it's somewhat maddening that very frequently you'll hear any particular trait or disease is perhaps 50% genetic and 50% environment. However, there represents a spectrum where certain traits such as psoriasis or depression can have very strong genetic influences that aren't modified greatly by your environment, and then things like cancers and multiple sclerosis, which are strongly environmentally influenced. Cancers, the environmental influence, the number one influence is whether you smoke or not. For multiple sclerosis, there is a clear correlation with geography. The further you are away from the equator, the more you are likely to have the disease, which has led people to postulate that some sort of infectious component is involved, and that the geographical climate for this infectious source, whether it's a bacteria or virus or other, does not thrive at certain geographical locations. Okay, thank you for listening.